Hey guys, in this video we're going to revise the structure of DNA, and we're going to learn a little bit more detail than we did when we studied it before in chapter 3. So I'm going to start sharing my screen with you now. It'll look a little weird for a moment, but then you'll be able to see our slides, which are under chapter 10 on Blackboard. So in this first video we're going to revise the structure of DNA. So let's take a look at what we're going to look at, our objectives here. We're going to describe the structure of the two nucleic acids, DNA and RNA. We're going to describe the notion of complementary base pairing. So complementary base pairing is, remember, A will always stick to T, right? So the A will always stick to the T, and the G will always stick to the C in DNA. And remember, in RNA, there's no T, so A sticks to U in that case. So remember that the bases pair up like that, and always A sticks to T and G sticks to C when you're looking at your DNA. And then we're just going to remind ourselves, what's the double helix model of DNA? What does a piece of DNA look like? The different ways you can model DNA. So first of all, where would I find DNA inside my cell? I would find it in the nucleus. So remember, we said the DNA of the organism is in the nucleus, but remember there's also some DNA in the chloroplast and the mitochondria, but that is not our DNA. That's the DNA of the microbes that were taken inside our cells. So our organism's DNA, our genome of our organism, is going to be inside the nucleus. So remember, the nucleus is in usually one of the largest organelles in the cell. It's got this nuclear envelope separating the nucleus from the surrounding area. And remember, there are pores, tiny holes in the nuclear envelope. So our DNA is going to be inside our nucleus. And remember, when our cell is not dividing, we're going to refer to this DNA strands as chromatin. And then if the cell starts to divide, the chromatin coils up and becomes chromosomes. Also inside your nucleus, there's an additional structure called the nucleolus. The nucleolus is where you're going to make your ribosomal RNA, and we're going to see later why we need ribosomes. So here's our nucleus, and it contains the DNA, the genomic DNA of the organism. So we've got two types of nucleic acids. We've got deoxyribonucleic acid, which is DNA, and ribonucleic acid, which is RNA. And they have different structures, which we talked about before, but they're all made of building blocks called nucleotides. So let's look at one nucleotide. What's found in one nucleotide? There's a sugar. So remember, if this is DNA, the sugar will be deoxyribose. If it's RNA, the sugar will be ribose. So this is a nucleotide from DNA. It's got deoxyribose sugar. Then attached to one of the carbons, we have the phosphate group. So remember, this was one of our functional groups. So remember, we said that DNA had a sugar phosphate backbone. So the sugars and the phosphates are all going to link to each other. So this phosphate will have another sugar linked over here, and so on. And then attached to the sugar phosphate backbone is going to be the bases. So remember, for DNA, we've got 4 base possibilities: adenine, guanine, cytosine, or thymine. So these are our nitrogenous bases. So on this particular nucleotide, the base thymine is attached, but you could have any of the 4 bases attached here. So these nucleotides are the building blocks for your nucleic acids. Now, the nucleotides could have different bases, like I said, and remember that in DNA, you're going to have A, T, G, and C, and then you only get this last one, uracil, when you're looking at RNA. So a big difference between DNA and RNA is that DNA has the bases A, G, C, and T, and RNA has the bases A, G, C, and U. So you never find T, thymine, in RNA. So there are 2 groupings for these bases. They are the purines and the pyrimidines. So adenine and guanine are purines. Cytosine and thymine are the pyrimidines. And the way I remember who's who is the pyrimidines have a Y in them. Cytosine, see, there's a Y there, and there's a Y in pyrimidines. thymine, there's a Y here, and there's a Y in pyrimidines. So that helps me remember. And then I remember that in RNA, we don't have thymine, so instead we have uracil, so these two have to be in the same grouping. And you'll notice that when you make your links, A connecting to T, a purine, adenine, is sticking to a pyrimidine. And when G binds up with C, a purine is sticking to a pyrimidine. So these are linking up with each other. So the purines also have two ring structures. See the two rings? So these are the purines, and the pyrimidines just have one ring, a one-ring structure. So when A sticks to T, a purine is sticking to a pyrimidine. The two-ring structure is sticking to the one-ring structure. Okay? Now let's take a look at the structure of DNA when it's all together. So remember the two guys, James Watson and Francis Crick, came up with this idea that DNA was a double helix. So a twisted ladder is how they've also described it. But remember, I told you in earlier videos that this lady, Rosalind Franklin, did a lot of the work and the X-ray crystallography that helped the other two realize the shape of the DNA. But unfortunately, she died before the Nobel Prize was given out for finding the structure of DNA. So she didn't get acknowledged, which is sad because she did a lot of the work. So let's take a look at our DNA. Here's the sugar phosphate backbone. See, this is a sugar. This yellow circle's a phosphate. This is a sugar. This yellow circle's a phosphate. So our DNA has sugar, phosphate in the backbone. And remember, this is DNA, so this is the deoxyribose sugar, phosphate. Deoxyribose sugar, phosphate. So that makes up your backbone, and then your bases stick in towards the center of the molecule. And remember, A is sticking to T and G is sticking to C. Now also here you can see that there are two connections between A and T. A and T, the bases, are held together by hydrogen bonding. So that's a very common multiple choice question. What holds the bases together? The bases are held together by hydrogen bonding. So between A and T, there are two hydrogen bonds. Here's one, here's two, right? Between G and C, there are 1, 2, 3 hydrogen bonds. So G and C has 3 hydrogen bonds holding it together. So we need to know that between A and T nucleotide bases, there's 2 hydrogen bonds made, and between G and C, 3 hydrogen bonds made. So this gives us our structure of a DNA. So it's kind of like a ladder. The sides are made of the sugars and phosphates. The rungs are made of our bases, A's and T's and G's and C's sticking together. So remember, the DNA molecules are double-stranded. They've got the sugar phosphate backbone. See these little 5-sided structures? These are the sugars, and then the phosphate links the sugars together. So you got sugar, phosphate, and here are your bases, your A's and T's with the 2 hydrogen bonds holding them together, and your G's and C's with the 3 hydrogen bonds holding them together. So the DNA molecule is a double-stranded double helix. A always sticks to T, G always sticks to C. So there's several ways you could look at DNA, and your book had this really nice picture. So you could look at it as the ribbon model, where you've got the kind of twisted ladder idea with the bases sticking into the center. Or you've got the atomic model showing you all the linkages and the bonding between the different structures. So by the way, between the sugars and the phosphate, this is a covalent bond. So these are covalent bonds. These are hydrogen bonds here. So between sugar and phosphate, these are covalent bonds. And remember, in covalent bonds, the electrons are shared between the elements. So the sugars here and the phosphates are sharing electrons. So that's how they're stuck together. So this is covalent bonding in the backbone. And then this is hydrogen bonding, holding the A's to the T's and the G's to the C's. And this is a computer model of what it might actually look like inside the body. So you can see it gets very complicated when you start looking at real life models. So that's the end of our first video, learning all about, just reminding ourselves about DNA. And when we come back, we're going to learn about the different names that we use for the different ends of the DNA and why that's important.